The ongoing challenge of feeding a growing global population in the face of climate change has intensified the search for innovations in agricultural productivity. Photosynthesis, the fundamental process driving plant growth, offers a promising avenue for enhancing crop yields. Most of the world's staple crops, such as rice, wheat, and soybeans, utilize the C3 photosynthetic pathway, which is less efficient in hot, dry, and high-light environments. In contrast, C4 plants like maize and sugarcane exhibit superior photosynthetic efficiency under such conditions due to specialized anatomical and biochemical features. Engineering C4 traits into C3 crops represents a groundbreaking strategy to improve photosynthetic efficiency, thereby increasing yields and enhancing food security. This documentary delves into the biological principles, the historical context of this research, current progress, challenges faced, and future prospects of engineering C4 photosynthesis into C3 crops. Understanding the rationale behind engineering C4 traits into C3 crops requires a comprehensive examination of the differences between these two photosynthetic pathways. C3 photosynthesis, the ancestral form of photosynthesis, occurs in the majority of plant species, including wheat, rice, and soybeans. In this pathway, carbon dioxide is initially fixed by the enzyme ribulose 1,5-bisphosphate carboxylase oxygenase, or Rubisco, into a 3-carbon compound, 3-phosphoglycerate. However, Rubisco is notoriously inefficient due to its dual affinity for oxygen and carbon dioxide. When oxygen is fixed instead of carbon dioxide, a process known as photorespiration occurs, wasting energy and carbon skeletons that could otherwise contribute to plant growth. C4 photosynthesis, by contrast, evolved independently in several plant lineages, and is characterized by spatial separation of carbon fixation and the Calvin cycle. In C4 plants like maize and sugarcane, carbon dioxide is initially fixed by phosphoenol pyruvate carboxylase, or PEPC, into a 4-carbon compound in mesophyll cells. This compound is then transported to bundle sheath cells, where carbon dioxide is released and fixed again by Rubisco in a carbon dioxide-rich environment. This spatial separation minimizes photorespiration, enhancing photosynthetic efficiency, especially under conditions of high temperature, intense light, and limited water availability. Research by Sage et al. In 2012 highlights that C4 plants are up to 50% more water efficient and 30 to 60% more nitrogen efficient than C3 plants under equivalent conditions. The efficiency arises from the high affinity of PEPC for carbon dioxide and the reduction of oxygenase activity by Rubisco. Consequently, C4 photosynthesis supports greater productivity, particularly in tropical and subtropical regions. The world's food supply largely depends on C3 crops, particularly rice, which serves as the primary food source for over half of the global population. However, the photosynthetic inefficiency inherent in C3 crops imposes a ceiling on yield potential, especially in tropical regions where photorespiration significantly curtails productivity. Predictions by the Food and Agriculture Organization suggest that global food production must increase by approximately 70% by 2050 to sustain the growing population. Concurrently, climate change exacerbates heat stress, drought frequency, and elevated atmospheric carbon dioxide levels, intensifying the limitations of C3 photosynthesis. Historical yield improvements driven by the Green Revolution predominantly stemmed from enhanced agronomic practices, improved genetics, and expanded cultivation areas. Yet yield gains have plateaued for several staple crops, necessitating novel interventions like photosynthetic pathway modification. For instance, Zhu et al. Estimated that transforming C3 rice into a C4 plant could boost photosynthetic efficiency by 50%, potentially increasing yield by up to 35% under optimal conditions. Engineering C4 photosynthesis into C3 crops requires a multifaceted approach, incorporating both anatomical and biochemical transformations. C4 photosynthesis is distinguished by the development of Kranz anatomy, wherein mesophyll cells surround bundle sheath cells, facilitating efficient carbon shuttling. However, most C3 plants lack this anatomical differentiation, presenting a significant obstacle for researchers. At the biochemical level, the conversion requires introducing genes encoding key C4 enzymes, including phosphenylpyruvate carboxylase, or PEPC, pyruvate orthophosphate dekinase, known as PPDK, and NADP malic enzyme, often abbreviated as NADP-ME. Early experiments by Kayala et al. In 2011 successfully introduced these genes into rice, demonstrating increased photosynthetic rates in controlled environments. Nevertheless, achieving coordinated gene expression, appropriate enzyme localization, and the development of Kranz anatomy remain significant challenges. Molecular studies suggest that the evolution of C4 photosynthesis involved alterations in gene expression patterns, rather than the emergence of entirely novel genes. Comparative transcriptomic analyses between C3 and C4 species reveal shifts in gene expression profiles of transporters, enzymes, and structural proteins critical for C4 function. This finding implies that manipulating regulatory elements might be sufficient to induce C4-like traits in C3 crops without introducing foreign genes. The International C4 Rice Consortium, launched in 2006, represents one of the most ambitious initiatives in this field. Spearheaded by the International Rice Research Institute, or IRI, the project aims to transform rice, a C3 crop, into a C4 plant to increase photosynthetic efficiency and yield potential. The consortium adopts a phased approach, starting with the identification and insertion of key C4 genes into rice, followed by attempts to induce Kranz anatomy and optimize metabolic pathways. One notable breakthrough came in 2015 when Wang et al. Reported the successful introduction of PEPC from maize into rice. The transgenic rice exhibited a 15% increase in photosynthetic rate under high light and temperature conditions compared to wild-type plants. Subsequent efforts focused on co-expressing multiple C4 enzymes and investigating regulatory elements that modulate cell-specific gene expression. Parallel research in wheat, another critical C3 crop, has also made strides. Studies conducted by Ermakova et al. In 2019 demonstrated enhanced photosynthetic performance in wheat after the insertion of maize PEPC genes. However, the improvements were less pronounced than in rice, reflecting species-specific differences in anatomical and biochemical constraints. Despite significant advancements, engineering C4 photosynthesis into C3 crops presents formidable challenges. One major hurdle is the development of Kranz anatomy, which involves complex spatial organization of mesophyll and bundle sheath cells. Researchers have identified several candidate genes associated with this anatomical trait through comparative genomics, but replicating the pattern in C3 species remains elusive. Another critical challenge lies in ensuring the synchronized expression of C4 enzymes across different cell types. C4 metabolism requires precise spatial and temporal coordination, as disruptions can lead to metabolic imbalances and reduced efficiency. Studies by Kovshov et al. 2016 emphasize the importance of promoter selection, as cell-type-specific promoters are essential for correct enzyme localization. Metabolic compatibility also poses difficulties. The introduction of C4 enzymes can inadvertently affect other metabolic pathways, given the interconnected nature of plant metabolism. For example, increased PEPC activity may alter nitrogen metabolism and impact amino acid synthesis. Comprehensive metabolic modeling, supported by systems biology approaches, is crucial for anticipating and mitigating such side effects. The successful engineering of C4 photosynthesis in C3 crops promises significant agronomic and environmental benefits. Enhanced photosynthetic efficiency directly correlates with higher biomass production and grain yield. Model-based projections by von Kemmerer et al. 2012 suggest that C4 rice could achieve yield increases of 30 to 40% under current agricultural conditions. Water use efficiency stands out as a particularly compelling advantage. C4 plants inherently lose less water per unit of carbon, fixed due to reduced stomatal conductance. In water-scarce regions, C4 engineered rice could sustain yields while reducing irrigation requirements, aligning with global efforts to promote sustainable agriculture. Moreover, C4 photosynthesis exhibits superior nitrogen use efficiency, a critical factor given the environmental concerns associated with nitrogen fertilizer use. Enhanced nitrogen assimilation in C4 rice could decrease fertilizer dependence, mitigating nitrate leaching and associated eutrophication in aquatic ecosystems. The introduction of genetically modified, or GM crops, including those engineered for C4 photosynthesis, inevitably raises ethical, socioeconomic, and regulatory concerns. Public perception of GM crops remains polarized, particularly in regions heavily reliant on rice as a staple food. Transparent communication about the safety, benefits, and potential risks of C4 crops is essential for fostering societal acceptance. Intellectual property rights also warrant careful consideration. Collaborative efforts like the C4 Rice Consortium emphasize open access research to avoid monopolistic control over this transformative technology. Additionally, regulatory frameworks must adapt to evaluate the unique characteristics of photosynthesis-engineered crops, ensuring rigorous safety assessments while facilitating innovation. Socioeconomic analyses highlight the potential for C4 crops to alleviate poverty and enhance food security in developing regions. Improved yields and resource use efficiency could reduce production costs and stabilize food supplies, particularly in climate-vulnerable countries across Asia and Africa. Ongoing research continues to explore innovative approaches to accelerate C4 engineering. Advances in genome editing technologies, particularly CRISPR-Cas9, have revolutionized the precision and efficiency of gene modification. For instance, CRISPR-mediated editing of regulatory elements in rice has successfully altered leaf anatomy, providing preliminary insights into the genetic architecture of Kranz development. Synthetic biology also presents exciting prospects. The design and construction of synthetic metabolic pathways offer the potential to bypass some of the evolutionary constraints of natural C4 photosynthesis. Researchers at the University of Cambridge have experimented with synthetic pathways to enhance carbon assimilation in tobacco, a Model C3 plant, demonstrating the feasibility of such approaches. Interdisciplinary collaborations between plant biologists, geneticists, agronomists, and computational modelers will remain essential for overcoming persistent challenges. Large-scale field trials supported by international funding bodies and agricultural institutions are crucial for assessing the performance and ecological impact of C4 engineered crops under realistic agricultural conditions. The engineering of C4 photosynthesis into C3 crops represents one of the most ambitious and promising frontiers in plant science. By addressing fundamental limitations in photosynthetic efficiency, this innovation has the potential to significantly enhance crop yields, resource use efficiency, and climate resilience. While substantial challenges remain, ongoing research continues to yield encouraging results, propelled by technological advancements and global collaborative efforts. In a world grappling with food security and environmental sustainability, the successful realization of C4 crops could usher in a new era of agricultural productivity, offering hope for a more secure and resilient global food system.